All right. Sorry, guys. A little dark. I'm afraid I only have lamplight tonight and no ceiling light. Still trying to get acclimated to the new place. So it looks like I missed everybody last week. I noticed that there was not a 9 p.m. spotlight session all week, and I don't know if that was a mistake because it was Labor Day week or it didn't get added. Because I kept logging in and I'm like, nobody came in, and I'm like, well, maybe it's exam week or something. And then I realized that IT did not. Yeah, yeah, and that I know, Tyler. So that was a little bit out of my hands. So I apologize. Um, but hey, I'm back. Looks like my students are preparing for exam one this week. I believe their window is open now. And then some of the other sections have either started or completed exam one, moving on to exam two or unit two. I'm not sure where we're at. Maybe we're doing a little bit of chapter three tonight. Where are we at? Why don't you all chime in here? Looks like we got some late ads. Welcome, welcome. Uh, Go ahead and chat me and figure out what chapter we're on. All right, good, Justin. That's all. That's what we're here for. All right, chapter three. All right. Let me get on the right screen here because it's telling me I got all these chats and I see them. Okay. So chapter three. What do we want to do? Chapter 3, 4, and 26. Okay. Thanks, Stephanie. Membrane transport. All right. Thank you, Hannah. Membrane transport. So I'm going to walk and talk because I'm gonna shut this door. Excuse me. So membrane transport really is passive transport, active transport, and diffusion. We won't talk about diffusion as well. We can touch on diffusion. We'll kind of build up, right? So diffusion. Okay. All right. Thank you. Thank you for being on the ball there, Justin, Emily, and Candace. You guys were. Thank you. So I don't know why that happens. It just happens, and you have to let me know. Another thing. I see some folks in here with just first name or some initials. It's very, very hard to give you attendance points if you don't put your full name in. So be sure to put your full name so computer people can find you in the system and give you attendance points for these spotlight sessions. Okay. FYI, there you can usually go away and then retype your name and then join. No, I got your first and last name, Emily. You're good. Just don't do anything else. Okay, so diffusion or concentration, really what we're doing is we're tracking solutes. All right, we are really kind of talking about solvent versus solutes, and solutes is something that we dissolve in, let's say, water. Typically, solvent is always going to be water. All right, it's going to be the more of something. In our bodies, it's water. So when we put solutes in, that's either salt or potassium, right? And that's like electrolytes. That's what we're really talking about when we're talking about membrane transport. Okay, we are really controlling the amount of electrolytes coming in and out of our bodies. And you sweat out salt. What you do when you drink, if you've noticed, if you've ever sweated profusely, probably by the end of the day and you drank a lot of water, you might find yourself craving like something salty, like some fast food. That's sodium central. So that's just because your body is a little bit low on sodium, and it's going to want a nice dietary deposit of sodium. So, Haley, I'm not sure why you can't hear me. My microphone is on. Try to turn your. Let's see. Everything of mine is on. I'm not sure why you can't hear me. Um, I would disconnect and reconnect. So when we're talking about diffusion, we're really following the concentration of solute from high concentration to low concentration. Okay, if we put some sugar in our coffee, the sugar is going to be solute. All right, you'll notice that eventually the sugar will dissipate down its concentration gradient. All right, another example would be putting food dye. Have you ever, like, let's say if you watch Jaws or you watched, some horror film and they have blood that drops in water? Well, what's it do? It kind of like disseminates out, right? That's because the blood or the food dye maybe is going from high concentration, which is, the dye itself or the blood drop, to low concentration, and it spreads out into the solvent or water in this case. So that's the concentration gradient that we're talking about. This is kind of what drives membrane transport. Okay, so that's the simplest of them all. Simple diffusion is, I think, as they call it. All right, and then we start getting into the membrane part. Right, we talked about transport, or the movement of solute. Now let's introduce the membrane part. So the membrane part, and we're going to build on this more, is when all cells are pretty much created by membranes, phospholipid bilayer. That kind of goes back to what we talked about in chapter 2. Hydrophilic heads or polar heads with hydrophobic tails or nonpolar tails. Water loving versus water hating. Water loving, the membrane, that's the polar heads that stick out, right? And you have the fatty tails on the inside. All right, that's how we separate and we can control what's coming into this cell. All right, like one hand is one side of the membrane and this hand's on the outside. In between is the non-polar part, water hating, and then on the outside's the water loving part. Okay, and then all of a sudden we have these channels that are like in between, right? And these channels are really what regulate electrolytes. Okay, so for example, it just so happens that we have a high concentration of sodium outside of our body. Let me turn my screen up so maybe y'all can see my face a little bit. His hair is a bit wild, but that's just because I cut the grass late. So when sodium is around the cell, on the outside of the cell, it is in high concentration. And what it wants to do, it wants to get into the cell. It wants to go from high concentration to low concentration. All right, so it will find a transport or a channel, a sodium channel, so to speak, and it will flow into the inside of the cell. All right, that's through facilitated diffusion. Okay, because we facilitate that channel when it goes in. All right, now that's an electrolyte. A sodium is an electrolyte. Now, that's the most common electrolyte. However, it's not the most important electrolyte. Does anybody know the most important electrolyte in your body? Anybody want to throw anything out? Yeah, that's glucose. Glucose is probably second, Bethany, especially with diabetics, right? People who have low blood sugar. It's not exactly sodium's close, Emily. Yeah, sodium's important. You're gonna want that, but there's another one. All right, and the element is K. Yeah, good job, Nicholas, Hannah. Yeah, you guys got it. So you know what happens when you go to what's it called? Capital punishment. where they give you the juice because you've committed an awful crime, and I can't think of it. Anyways, the concoction that they give you is a large bolus of potassium. Okay. Now you may think, well, why would somebody get a potassium into their system? Well, it just so happens that the potassium levels in your body are highly regulated. Okay. It's many oftentimes when people go into kidney failure, their potassium can sometimes build up in their system, especially if they're dehydrated, like old people. And the more potassium you have in your body, the more likelihood it can slow your heart rate and almost stop your heart, either where you pass out or you just, your heart just stops beating. And that's what happens during capital punishment. Um, I'm not sure if that's the best word to use. I'm trying to think. It's not the electric chair. I guess it's just capital punishment. Um, and well, opposite, Bethany. Yeah, lethal injection. Thank you, guys. Thank you. That's what it is. Yeah, thank you, Nicholas and Allison. I appreciate that. Some things just I can't get a grasp on. Yeah, but back to your point, Bethany. Potassium, when potassium gets low, sometimes it can almost cause your heart to go into arrhythmia. All right. And sometimes they'll can give you like a bolus of potassium. It has to be highly regulated, okay, to slow your heart rate down a little bit. I'm not sure if they would get, like, let's say somebody went on like an overdose of cocaine or some kind of maybe methamphetamine. I'm not sure if they would give potassium to slow the heart, but they could. Because then you're kind of mixing uppers and downers. Okay, you're mixing stimulants with depressants. All right, and sometimes that can throw the arrhythmia off on the heart. But I think in some severe circumstances, I think potassium has been used to slow the heart. It just has to be very carefully concentrated. Okay, because if you do it too much, yeah, if you do it too much, you can stop the heart and you're in trouble. So potassium, when I said is highly regulated. Okay, so it just so happens that it's not a lot potassium, but the more concentrated this little bit of potassium is, it's inside the cells. Okay, inside the cells, there is a higher concentration of potassium inside the cells. Quite opposite from what I just told you with sodium. The higher concentration of sodium is on the outside. So sodium wants to get in, and then potassium wants to get out, okay, because it wants to go from high concentration to low concentration. All right, so sodium's coming in, potassium's going out. If y'all can follow my hand gestures here, it's the best I can do tonight. So we have different membrane transport channels that are solely dedicated to regulating potassium and sodium. All right, we're going to throw chloride in there too. All right, we'll talk about chloride next. It's a minor player. So all of a sudden, have we talked about action potentials yet in class? Okay, so we kind of know what I'm getting at, right? So all of a sudden, if my brain sends an electrical signal to my muscles or to my mouth, moving my mouth muscles so I can speak, what's really happening is that we're getting these action potentials sent down to the muscles, which is controlling these channels of sodium and potassium triggering at the same time, which causes this membrane transport. Yeah, Allison, I'm getting to that. We're going to talk about that. Because this is how, this is how we reset, right? When we reset, when we have all of a sudden the sodium comes into the cell and potassium goes out of the cell and it's done, we don't have enough to recoup the next action potential. So what we use is called, the next idea is called active transport, active membrane transport. All right, and the part that's active about it is that we have to use ATP to reset it, okay, to reset these concentrations. Well, you may ask me, well, what do you mean reset? Well, what I mean is that we literally have to pump this sodium back out of the cell and pump the potassium back into the cell. All right. Sodium gets pumped out, potassium gets pumped in. And what that does is that restores these concentration gradients. So when the next electrical conduction fires, then we have these ions that could all of a sudden go down their concentration gradients through these membrane channels and carry out another action potential. Okay. So Allison, so what does it mean when it talks about sodium potassium pump? Like how sodium comes in, potassium wants to go out. Well, I think I just explained it, but I'll hit it again. So we use ATP. Remember we talked about adenosine triphosphate a couple weeks ago when we talked about macromolecules and nucleic acids and how adenosine triphosphate has all this energy packed up into this molecule because it has a bunch of phosphate bonds right next to each other. And when you put a bunch of phosphate bonds right next to each other, there's so many electrons on those oxygens, right? P, phosphorus is the P, O3, 3 oxygens around that phosphate. Each one of those oxygens has a bunch of electrons around it. You stick a bunch of electrons in a small space, all of a sudden you got a lot of energy. You got a lot of like negative tension, right? That wants to like snap off and kind of fly off because you got all this negative charge packed up. But like repels like, right? So all of a sudden, when you break that bond, you get a little bit of energy released, and that's why we use ATP. We take this energy and somehow through other means that we won't dig into, it allows the sodium to get pumped back out of the cell. We will pump 3 sodiums out for every 2 potassiums in. That's usually a exam question, okay? 3 sodiums out for every 2 potassium in. So this brings me to my next point. Do we know about membrane potential? Have we talked about membrane potential? Okay. So just humor me on a little bit, Hannah. Why do you think membrane potential on a resting cell is negative? Because if you remember, we kind of start negative. We start probably about minus 80 millivolts, okay? Minus 60, it's actually minus 80, okay? Minus 80, minus 70 millivolts. And you're like, well, what is a millivolt? Well, that's basically we're measuring the current. We're sticking an electrode, which is a measuring rod, into this cell, and we're measuring the voltage or the it's not current. I guess it's just electricity or voltage inside the cell, and it just so happens to be negative. So you're probably maybe scratching your head at this point because I feel like if you had an idea, you might have shared it. But I'll just I'll go on here because it's a little bit of a tough question, but I'm about to connect the dots here. I told you that we have 3 sodiums going out. Okay, so what is sodium? Sodium is a cation, right? From chapter 2, cation, positively charged, right? So all of a sudden we have 3 positively charged sodium ions going out of the cell. All right, and then we're only bringing in a potassium. But potassium's also a cation. So we're bringing in only 2 potassiums, right? So 3 cations are going out, but only 2 cations are going in. Yeah, you're right. The inside is less than the outside. So all of a sudden we have this negative charge because we have all these cations that are, getting kicked out. And it just so happens that, well, you might say, well, that doesn't really explain the negative part. That just explains the flow. I want to dive in a bit deeper, which you don't have to know this, but just for the sake of completeness, phosphate, PO3 minus, all right? Phosphate has a negative charge on it just because it just does. If you can draw it out and you'll see. So you may realize in chapter 3 that DNA has a hell of a lot of phosphate in it, okay? Connected to the nucleotides. That holds a lot of negative charge inside the cell. That's why the cell is more negative. Okay. Um, I'm sorry, Cassandra. Was that something towards me? You lost track of time. Help clarify that if you want, and I'll see if I lost you on my explanation. So that's the resting membrane potential, right? Because we have, pretty much what's controlling the resting membrane potential is the sodium potassium active transport. Um, Sodium potassium pump, otherwise known as an active transport. All right, so then all of a sudden we get this electrical conduction coming into the cell, which opens up all these channels, right? Oh, you're fine. Don't worry about it. Welcome to the spotlight. Um, when you have this electrical conduction come on and it wires up all these transports, that's when you get sodium rushing in, potassium rushing out. But we get more potassium rushing in because if I told you that's a much higher concentration, and that's why you get the membrane potential shoot up to like plus 20 or plus 30 millivolts, and then, goes back down. It gets a depolarization going up, and then it repolarizes. And when it repolarizes, basically what that is that that's just it's it's the potassium channels that are a little bit slower in opening, and then as the potassium rushes out, all that positive charge rushes out of the cell, and it brings the membrane potential back down to resting, only to be restored by the sodium potassium pump, ATPase pump. So I'm going to type something in here. All right, so I just gave you the ATPase pump. Whenever you see this, um, whenever you see this ACE suffix on something, ATPase, that means that we're really talking about an enzyme. Okay, so as you recall, gotcha. All right, sorry. Thank you for catching that early. Um, it's nice to see you guys are paying attention because there's been past semesters where I could go on for minutes and I'll never know because I'm in the I'm on a tear. So let's back up. When we see this ATPase, this ACE suffix, that is an enzyme, right? Well, when it's an enzyme, what that really is that means it's just a protein carrying out a reaction, right? So anything with ACE on the end of it is an enzyme carrying out a reaction. In this case, it's pumping out sodium and pumping in potassium. That's all that is. So you may hear me refer to the sodium potassium ATPase pump. That's just an active transport that's restoring the resting membrane potential of the cell. All right. So is there any questions there? Almost, Hannah, almost. So when you reach equilibrium potential, that really is when, so a question on an exam would be, what what ion, what electrolyte controls the resting membrane potential? Well, the only one that's really controlling it is the potassium, because for other reasons, potassium there's such a high concentration of potassium that wants to get out of the cell that it actually leaks out of the cell. And so sodiums are really controlled. So the sodium transports are controlled. Uh, there's still have a little bit of flow, but probably about the resting, potassium leak channels. Yeah, you're right. So the resting membrane potential, if it was only potassium channels, you would be considered at equilibrium potential. Basically where the concentration is equal, the chemical concentration is equal to the electrical concentration. I think that's the definition of equilibrium potential. So uh, I think it may be you, Haley. I'm sorry. My microphone's working. I'm not sure why you can't hear me. That's a bummer. Um, but try to reconnect and come back in. I'm not sure what it is. It is recorded. We can put it up on YouTube for other purposes. So when when the sodium plays a little bit of role in that resting membrane potential, that really takes it off of the equilibrium potential, because you may have about 98% of the potassium channels controlling the resting membrane potential, and maybe about 2 or 3% of the sodium uh, channels working. So that's why your resting membrane potential may be at negative 86, negative 85, and the potassium equilibrium potential is like minus 90. Does that answer your question, Hannah? That's some pretty elevated physiology. That may be a little bit beyond the scope of this class, maybe not. Um, okay, yeah. So that's why the resting membrane potential never really reaches negative 90. It's always like negative 88, negative 87, because you do still have probably about 2 to 3% of that sodium channel still kind of moving sodium around. It's not absolute. So yeah, good question. I always like physiology over anatomy. I think you can, if you get really good at physiology, you can tie in pathophysiology, you can tie in everything. So let's do some pathophysiology real quick. And I don't want to go too deep because I have to brush up on this myself. So I mentioned chloride, right? Chloride is what? Chloride, what charge does chloride have? If you look, yes, thank you, Hannah. Chloride is on the right-hand side of the periodic table. Chloride is also considered a halogen. Chloride, therefore, since it is a halogen, which is almost right next to the noble gases, holds about 7 electrons, right? So what the hell is keeping chloride from grabbing an electron from somebody else to fulfill its octet, right? I'm gonna have you all build up on some chapter 2 real quick. So if you got little old sodium over here who's in the first column or second column, first column, chloride will go find that cation sodium and rob it of its only electron. That's how you get Na plus. Okay. So then all of a sudden you get what? Table salt. What's table salt? Table salt is NaCl, right? I just typed it in. Na positive Cl minus. That's why it goes together. Anytime you see sodium, you're likely going to have chloride right behind it, just because. That's why sodium always follows chloride. So when we talk about potassium, or excuse me, when we talk about chloride in pathophysiology, there's one disease out there that is a genetic inherited disease, and it's one of the worst ones out there, in my opinion. And you've always probably seen fundraisers for it. Unfortunately, it affects the people that end up having this mutation do not live. They probably live maybe in between their high teens to low 20s. Does anybody know what disease I'm talking about? I've mentioned it before in some of my spotlights. Some of you all might have been on that one or not. Ooh, Justin, good. It's not Lou Gehrig's ALS. I actually did my thesis defense on ALS. That is the motor neurons, the killing off of motor neurons in the upper spinal tract. That's why people tend to choke and aspirate and get pneumonia and die. No, this Lou Gehrig's, sometimes you can live into your 40s, 50s, 60s with ALS. Yeah, it's Nicholas, you're right. CF. Good call there. So cystic fibrosis is basically governed by a dysfunctional chloride management in the lungs. All right. And I'm not sure if it sometimes it originates in the kidneys, but most of the time it's in the lungs. And what happens is that you get this really thick, with the amount of chloride that builds up, it also brings in sodium. And somehow you get this real thick mucus that builds up, and it'll accumulate in the lungs. And that's why that's the fibrotic part. Okay, is when your lungs kind of end up drying out, and they get more fibrotic. And what that means is that you can't really breathe very well. You know, like emphysema basically means is when your lungs can't contract, right? They're too expanded. That means you really can't blow anything out. They're too large. Fibrosis of the lungs means you can't take a deep breath in and expand your lungs. Okay, and sometimes the best way to treat cystic fibrosis, or at least back in the day, is where doctors or physicians would really take their hands and smack the back of the patient's middle of their back, lower back, higher back, to just knock the mucus off their lungs so they are able to cough it up and exude it, okay, spit it out. And that's kind of how they maintain this mucus. It's all from one genetic mutation in the DNA, and which causes a dysfunctional chloride transport, which does not manage the chloride very well, and we get this inundation of chloride, which brings in salt, which brings in, you know, dried up mucus, and this is what happens. Yeah, that's kind of a rudimentary explanation, Candace. It's been a while since I've really dug into the nitty gritty of CF. I think it's up there with ALS and Alzheimer's, in my opinion. Hopefully, new technology out there will be able to correct this in children. There is technology out there where they can do genetic modifications. And if people have it, that they can really find a way to correct this and cure CF. I think it'll be in the next 5 to 10 years, honestly, if everybody just kind of backs off and lets science do its thing. Okay, so to finish up, and unless we have more topics, I'm going to introduce 3 more topics of membrane transport, and that's just easy vocabulary, is symporter, antiporter, and uniporter. Okay, so a uniporter is really just a transport that allows one electrolyte to go one way. Okay, either in or out, one of them. Symporter, sim means same. So, excuse me, what that really is when you have 2 ions going in or 2 ions going out. All right, they're going the same direction. Symporter, same direction. And then antiport is really just the sodium potassium ATPase pump. All right, where one's going this way and the other one's going this way. All right, anti means different. Antiport, a good example of that would be the sodium potassium pump. Okay, I think that finishes up my. I gotta sit back a little bit. My back's getting tight. I think that pretty much sums up what I want to talk about with membrane transports. Is there any other topics that we want to go over with chapter 3? That's DNA replication, transcription, translation. We have the rest of the week to go over this if you want. I've probably thrown a lot at you all tonight, so don't feel wigged out. We have to we have Tuesday, Wednesday, and Thursday. We're still not going to do a Sunday session. I haven't heard anything about that yet. And if you want me to, I can always throw this up on the YouTube channel if you want to rewatch it for whatever reason. Okay. 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 All right, guys. Yeah, I can do that. So, so just Google I gotta kind of knock the rust off and get the, yeah. So it's like Google, like Bio 137 Mattmiller Spotlight session. It's gonna be something like that. My last name and Spotlight and Bio 137. You should see it. I really don't know what my channel is. It's like maybe it's Andrew Mattmiller, Drew Mattmiller. I'm pretty basic. Yeah, I can do that. All right, guys. Cool. All right. Yeah, you can watch it. I'll try to disable the comments because I get dirty comments on there from bots. I think they're bots. Um, so I'm gonna try to avoid that. Okay. Um, yeah, no problem. Sounds like we're gonna wrap up tonight. I don't think you all have anything else we want to talk about, right? Okay. So I will. Yeah, no problem, Bethany. Nicholas, you too. I will catch you guys later on. I'll do the attendance. I need all the help I get. That's okay, Cassandra. Just kind of come with questions and concepts you want to go over. Okay. Once you go over some stuff, you gotta, if it's something doesn't sound clear, you gotta talk to me about it, and then we can work through it that night. And hopefully that will kind of help build on exam 1. So, all right, Justin, we'll see you Wednesday. All right, guys, I'm out of here. I will look for that YouTube thing in the next 20 minutes, okay? It's got to upload and download and all that. Takes some time. So, all right, take care. We'll catch you tomorrow night, if not later on in the week.